Hello everyone, good morning to you, and please get out your pen and paper because today's topic we are going to be talking about prokaryotic cells. This is a review of stuff I've learned in my microbiology class, and there's going to be a test later on this week, so I just wanted to make like a little study guide video thing for myself. And for all of you, if you find this information useful, please also stay tuned as well. Alright, so prokaryotic cells. So again, talking about microbiology, we are going to be looking at prokaryotes mostly, but we are also going to touch on eukaryotic cells as well because some of our microbes are also protists, which are usually microscopic. Okay, so first, we have the cell wall. We have our bacteria. Right here, we'll have a nucleoid. Since they don't have a bound nucleus, this is where we're going to find the DNA, inside the nucleoid. Also have pili, which are these tiny hairs outside the bacteria. We'll also have a cell wall, usually of peptidoglycan. And then, of course, our flagella. We also have some other layers and structures, such as the cytoplasmic membrane, as well as a capsule, but we're going to start off with the cell wall of bacteria. Right, so gives the bacteria strength due to its rigid structure. It's basically giving it reinforcement, kind of like how a plant cell wall would give the plant cell some reinforcement. Also gives it shape. And it's also an antimicrobial target. So many of these drugs, including penicillin, will actually target a certain type of cell wall in the bacteria. Speaking of which, we'll have our gram-positive and then our gram-negative. As a review from the last few times, gram-positive, they have a thick peptidoglycan layer. And peptidoglycan, it's only in bacteria. This is a substance you'll only see in bacteria. Notice I didn't say archaea, because archaea have something different. They have something called pseudopeptidoglycan, but bacteria in general have some peptidoglycan. Gram-negatives have an outer membrane. And when we use penicillin will affect gram-positive more than it will affect gram-negative because they rely on that thick peptidoglycan wall inside their structure. The structure of peptidoglycan itself has these alternating subunits. And you'll notice everything kind of has a pattern here. We have something called NAM and something called NAG. Now these are glycans, so they have a lot of sugars associated with them. This is N-acetylmuramic acid, and then this is N-acetylglucosamine. So we have some sugars in these structures, as well as some acid, as well as some, it looks like we also have some nitrogen. Amine, they have nitrogen. So these are glycans. This is from the word peptidoglycan. You see how it has pepti. This indicates protein or peptide, and then glycan will indicate that there are sugars present. And so if we draw this out, let's use some colors here to distinguish them from each other. We'll use that for the muramic acid one, and then for NAG, or the acetylglucosamine, we'll use red color. Okay, so we have our nag kind of in an alternating fashion like this. Actually, I made it too long. That's fine. Then we have our nam again, and then at the bottom, we're going to also have alternating nam and nag. Alright, so in between, we have something called a tetrapeptide chain. So from this nam, we're just going to have 4 of these tetrapeptides. So a peptide, again, think of it as an amino acid. So I'm just going to put 4 A's, like so. And this tetrapeptide chain links the glycans together. It gives it strength. The tetrapeptide here reinforces the structure. And then we'll also have this kind of a side chain here, a peptide interbridge. So 1, 2, 3, 4, 5. And this is only seen in gram-positive. This is again attributing to the strength and the thickness of that peptidoglycan layer seen in gram-positive. It's so thick and it's so supportive. And again, an additional link, such as this interpeptide bridge, is only seen in our gram-positive. Alright, so now we go back again to a tetrapeptide chain, and I'm going to go off, and again, tetrapeptide means 4 peptides, so 4 A's, 4 amino acid. Alright, so that's basically the structure of peptidoglycan. Now let's look at the membranes and the cells of a gram-positive and a gram-negative. So let's start with our gram-positive. So at the base, this is our basal layer. We're going to have that phospholipid bilayer again that has been covered many times, I bet, in science classes that you've taken, where we have the polar outside and then we have this nonpolar inside with those fatty acid tails, and then sometimes we'll have some proteins here, some integral proteins or a channel protein. So those are going to be represented by those 2 Subway sandwich $5 foot long shapes at the bottom. Right, next, so this is our membrane, which we've started afterwards, so cytoplasmic membrane. Next, we have a gel-like layer. So in order to show it's a gel-like layer, I'm just going to add like these squiggles. You know, in Jell-O, you kind of see all these air bubbles and squiggles, so that's what that's going to represent. Next, we have our peptidoglycan. So pep, and look how thick it is. So again, it's going to have these cross links between the glycans from the tetrapeptides. So I'm going to draw it kind of like a brick wall. Look at this wall. This is a very beautiful, tremendous peptidoglycan structure seen in our gram-positive. And it's extremely thick, and again, it has all those cross links reinforcing it. Now, if we look at our gram-negative, let's do it on this side in pink. So we still are going to have that phospholipid bilayer, or the cytoplasmic membrane. So I'm just going to draw a few of those phospholipids. Okay, now, it's interesting. We have quite a few different structures. So after the lipid bilayer, we're going to have our periplasm. This whole section is periplasm. But within that periplasm, we have this very thin layer of peptidoglycan. This is our pep. It's extremely thin. Next, after that, we have these giant, towering proteins called lipoproteins. So I'm just going to put lipo. And they're again giving structure. And then we have another layer. We have another bilayer. So We're going to just draw that in. These lipoproteins will go into this second bilayer. This is the outer membrane that we were talking about earlier. So again, another lipid bilayer with those lipoproteins going through it. This is the outer membrane, which is unique to gram-negative bacteria. Next, we'll have this weird chain of hair-like proteins. That's all I could describe them as. These are called lipopolysaccharides. So LPS, lipopolysaccharides, are those hair-like structures at the top. And each of those is a porin protein. Porin. So you can see a lot of differences between the gram-negative and the gram-positive. A little bit more on the gram-negative, that outer bilayer called the outer membrane is actually an endotoxin. A lipid that's part of it, called lipid A, is actually recognized by the immune system. And this will set off all different alarms because of the O antigen that will be actually recognized. And due to it still being a lipid membrane that has that inner nonpolar part, small molecules, as well as ions, will still go through those porins. Again, those are those hair-like proteins here. These porins that the small molecules and ions will still pass through. And if we have small levels of the lipid A or the O antigen detected, this will trigger the response in the immune system for gram-negative. It's going to set off those red flags, and we're going to start targeting them. So large amounts of the lipopolysaccharides, this will cause shock. And so it's kind of like an overload. So it's a little bit of a red flag, and it's also a warning to your body. So it's going to start using the systems of the immune response in order to target those gram-negative bacteria that shouldn't be there. Okay, so another interesting thought for gram-positive. So again, we're talking about gram-positive, so I'm going to be using this purple. So the gram-positive bacteria are actually only in the intestines. So when they are found, there isn't a specific response. It's more of a non-specific response from the immune system. So some responses that we have to the gram-positive bacteria include penicillin, which we've covered earlier, but penicillin, it stops the peptide bonds of the peptidoglycan from forming. And it actually prevents the cross-linking. So what it targets are these peptide bonds here. It prevents the cross-linking. That's why it's so effective against our gram-positive. And these cross-links are usually disulfide bonds. They aren't hydrogen bonds, they're disulfide bonds. They're extremely strong between these peptides, so it's able to break that. This handy drug or antibiotic is able to break down these gram-positive bacteria. the outer membrane of the gram-negative actually blocks penicillin. So this blocks penicillin. It just blocks access to this thin peptidoglycan layer here, so it doesn't really affect gram-negative. We also have lysozymes, and these break the glycan chains. So, instead of penicillin targeting the tetrapeptides, when we use a lysozyme against a gram-positive, it actually just kills off those glycan chains. So, with that, there we go. We shot a bunch of lysozyme at the peptidoglycan, and it was able to destroy all those glycan bonds. And this enzyme is actually found in saliva as well as tears. Alright, now let's go and talk about gram staining. So, crystal violet, the dye that we covered in the last episode, it actually stains the inside of the cell, not the cell wall itself. And in a gram-positive cell wall, so the crystal violet iodine complex is prevented from being washed out. So remember our primary dye, and then iodine. That was our mordant when we were talking about the process of gram staining. So this will stay in the PEP layer, and it doesn't get washed out. The alcohol that is added in the last step, the second to last step I should say, the decolorizer, alcohol will dehydrate the thick peptidoglycan layer, so it acts as a barrier. So now that dye is going to be stuck inside the cell. Not in its wall, but in the cell itself. That alcohol dehydrating the peptidoglycan layer really keeps it in there. Alright, now let's talk about the negative cell wall. The alcohol will now damage the outer membrane. And this exposes that thin layer of peptidoglycan, but it cannot retain the dye complex. That's why we have to go in with our safranin, and then to stain those gram-negative bacteria, they're going to be, again, pink. This is why we use safranin, because the crystal violet iodine complex won't be retained in the thin peptidoglycan layer. So that's why I use a counter stain like safranin. Alright, let's talk about bacteria without a cell wall. Yes, these do exist. It's very surprising. This was new info for me, but still interesting nonetheless. So the species in question is Mycoplasma, and they have a variable shape called pleomorphic. They don't have a shape, nor do they have a cell wall, so penicillin and lysozyme don't work. If Mycoplasma was Superman, this is like coming at Superman without any kryptonite. Penicillin and lysozyme won't attack Mycoplasma bacteria because they don't have a cell wall to begin with. However, they do have some cytoplasmic sterols, like cholesterol and whatnot, and these increase strength. So they still do have strength in their membranes. It's just because of these sterols. Alright, let's talk about archaea. Do they have cell walls? And actually, they do have cell walls. However, they have adapted to a wide range of environments, so they do not have they don't have any peptidoglycan. Sorry about that, I was getting my words fumbled all together, but they do have a similar molecule called pseudopep. So it's almost like peptidoglycan, but pseudo meaning false. It's not quite peptidoglycan. They also have S layers, and these self-assemble. Alright, I think that's all the professor wanted us to focus on that, so I'm going to go move on to capsules and slime layers. Alright, so this gives the bacteria the ability of invisibility. It gives them the powers that Invisible Woman would have. And they have this invisibility from the immune system because the antibodies cannot recognize or bind to the surface. Alright, so they are in general, they are a gel layer on the outside of the cell wall. And the capsule, it's more distinct and gelatinous, hence it's a gel capsule. The slime layer is, however, irregular, and it's somewhat diffuse. It's not as organized as our capsule. Most of them are made out of glycocalyx, a sugar shell, and then some of them are also made by polypeptides. And then the function is adhesion. When they could stick onto a surface, this allows them to grow biofilm. And this allows them to evade, again, the immune system. Gives them that invisibility that we were talking about earlier. Plaque is actually a version of biofilm. So a biofilm, again, is a community of multiple species of bacteria. They're all processing the nutrients in that one location. They adhere to it because, you know, there's a lot of nutrients. They like being around all those nutrients, so they're going to adhere to that surface. And then they make that biofilm. This calls in other bacteria who also want to join the party, and they live in this big old community. They keep adhering to the surfaces, and they just make this community where they're synthesizing compounds that others might need. They're evading the immune system. Likewise, if I had a drug that was targeting the bacteria in this biofilm, the ones at the top are going to be killed, so I'm going to have gravestones here. The ones at the top are going to be killed. However, the ones at the bottom are going to be safe because of that biofilm. It gives them protection. Again, it helps them evade the immune system. Alright, now let's talk about flagella. Again, the main point of flagella is movement. A ton of movement. Bacteria will do this movement where they'll like go in one direction, then they'll kind of reorient themselves and then go into the other direction like so, and then back and forth. It's kind of like this movement here to get to a certain point. It's not point A to B at all. It's kind of a jumbled mess. If this is like point A, then they'd actually go up and then down, and then they would get to point B. It's not a direct line. Alright, so let's talk more about flagella in general. It's a propeller movement, I should note that. And we have different ways to characterize bacteria. So we have monotricious. And again, we're going to classify the bacteria by the placement and the number of flagella. So monotricious, mono means one, so they only have one flagella. Paratricious, this means it's distributed all over the entire surface of the bacterium, like that. We also have lophotricious, and this means we have many on one side, so kind of like this little guy down here. And then we also have amphitricious, which means we could either have one on either side or multiple on either side. And then again, it's just propeller movement, helps them go to the places that they would need to find nutrients. Sometimes they do cause disease using their flagella as well. There are three parts to a flagella. We have the basal body, kind of looks like mechanical sort of object. So this is a basal body. Then we have our hook. And then we have our filament. And then it's made out of flagellin proteins. Let's talk about another part of bacterial movement, which is chemotaxis. It's where they sense chemicals, and this determines their movement. So nutrients attract the bacteria, while toxins will, of course, repel them. And then it's, again, run and tumble. We also have other types of taxis, or taxis, actually. But it's kind of like bacteria taking a little journey on a taxi, but the taxi is their flagella and their own body. So we have other types of taxis, too. We have aero. And this is if the bacteria are aerobic. And, of course, they'll go towards O2, or oxygen. We also have bacteria that are kind of like magneto. They're using magnetotaxis. Magneto of the X-Men, he has properties where he's able to sense the magnetic fields and interact with different magnetic poles. Well, some bacteria actually have little shards or fragments of magnetite. And this helps them orient themselves according to electrical fields and magnetic fields. And it's very interesting. We also have thermotaxis. And of course, this is for heat. And some bacteria do photosynthesize, so we also have phototaxis, which is going towards light. Another structure that bacteria have are pili, and these are shorter than flagella. And then if they use these pili to attach to a surface, this is called fimbriae. They give them the ability of twitching and gliding motility. And also there's a special type called a sexpilus, which a bacteria will extend to another, kind of like this, and they'll transfer DNA. This is like transfection or all those other methods that transform bacteria. This is another method where they're using a sex pilus to transfer a plasmid, which may give antibiotic resistance. This is a large part of antibiotic resistance, the bacteria transferring their plasmids one to another.